It's really my pleasure to introduce our guest speaker for today's NOCKS seminar. We have Dr. Shana Kelley, who is the Neena Schwartz Professor at Northwestern in the Department of Chemistry, Biomedical Engineering, and Biochemistry and Molecular Genetics. The Kelley Group works in a variety of areas spanning biophysical, bioanalytical, chemistry, chemical biology, nanotechnology, and has really pioneered new methods for tracking molecular and cellular analytes with unprecedented sensitivity. Dr. Kelley's work has been recognized with multiple awards, too many to list everything. I'm going to list the ACS Inorganic Nanoscience Award, the Steacie Prize, the Alfred Sloan Research Fellowship, an NSF CAREER Award, a Dreyfus New Faculty Award, and the naming as one of the top 100 innovators by MIT's Technology Review. Dr. Kelley is a Guggenheim Fellow, a Fellow of the Royal Society of Canada and the American Institute of Biomedical and Medical Engineering. Her work has been extensively cited with more than 80 papers, cited at least 80 times. She's an inventor of more than 50 patents worldwide. She's a founder of 4 molecular diagnostic companies. I'm going to list GeneOhm Sciences, which was acquired by Becton Dickinson in 2005. Dr. Kelley serves as a board director for the Fight Against Cancer Innovator Trust and the Canadian Academy of Health Science. She's an associate editor for the ACS Sensors and editorial advisory board member for the Journal of the American Society of Chemical Society, Nano Letters, and ACS Nano. So it's really my pleasure to introduce our special guest for today's seminar, and I will pause here.
All right, well, thank you so much for having me. I really appreciate the opportunity to tell you a little bit about some of our work. And as you've surmised from my introduction, I am a kind of physical scientist slash engineer, definitely not an expert in the field that many of you work in. But what I'm going to do today is tell you about a technology that my group developed and is working with. And this is one that we think hopefully will have applications to areas of interest for all of you. So I'll walk you through some of the work that we've been doing, and would love to hear your ideas about how we can continue to apply this in important areas. In general, my group, we create new technology platforms. We try to identify unmet needs in the life sciences or clinical medicine, and then really try to create typically new measurement platforms, technologies that allow us to make measurements that maybe we can't right now. And then we typically take those out and apply them very broadly. Historically, we've done a lot of work in the development of sensors, so electronic elements that can pick up on molecular analytes with a high level of sensitivity and specificity. And we've turned those into pieces of clinical diagnostic instrumentation. Increasingly, we're working on wearable sensors and real-time biochemical monitoring through the use of implantable and wearable devices. We also do some work on delivering molecules into cells using a delivery platform that is based on peptides. We've been working with that for several years. The area that I'm going to talk to you about today is this one right in the middle, which is the development of an approach for rare cell profiling. And I'm going to tell you about how we have applied that to the discovery of new therapeutic targets. We have some other applications as well in the therapeutic development space. So that's where I'm going to try to focus in today to keep this kind of on a single topic. I'm going to take the first few minutes and talk to you about just the development of the technology platform, this rare cell profiling approach. And so this will be a little bit more on the engineering side of things, just talking to you about how we think about these types of systems and how we eventually build them up. And so here, what we set out to do was to develop a new approach that would allow you to profile single cells, but to do so very quickly and efficiently so that you could look at large collections of cells and be able to identify interesting rare cells. And I'm sure, everybody who's here today is quite familiar now with everything that's happened in single cell omics and the incredible things that we can do now in terms of characterizing the genomics or transcriptomics or even the proteomics of single cells. But there are many applications where you're kind of below the noise floor of those types of approaches. And you simply don't have enough cells to really power those types of technologies. So that's where the approach that I'm going to tell you about today comes in. And the kinds of problems where this capability can be relevant, again, it's rare cells. It's things like circulating tumor cells that have broken off of tumors and are circulating around in the blood. And if you were to isolate a milliliter of blood, even from a patient with advanced cancer, they might have 10 or 20, or maybe if there's high levels in a sample, maybe 50 circulating tumor cells in a milliliter of blood where there's billions of normal blood cells. So again, these cells are really needles in a haystack. Same thing when it comes to tumor reactive T cells. So if you look at the immune cells that end up in tumors or even in the circulation, there's just a very small fraction of them that are tumor reactive. And so you need to, again, have the ability to sort through many, many cells to find ones that are actually educated about the molecular level features of a tumor. And then the work that I'm going to focus on today actually we'll hone in on using this rare cell profiling capability to look at CRISPR edited cells. And now we're quite easily able to make libraries of cells where we use CRISPR-Cas to knock out a single cell or a single gene, in a cell. We can have large collections of these cells, but how do we sort through them to identify phenotypes of interest? So that's the general problem that this platform was pointed at. And again, it's really to make this type of search for a rare cell very efficient. Most of the single cell technologies that we have access to today are serial in that you look at one cell at a time. And again, if you have to look at 1 billion cells to find 10 or 20 that are of interest, it's going to take you a very long time to do that. It's like looking for a needle in a haystack and doing that by looking at every single piece of hay. And so we want to come up with approaches that are much more efficient. And so, for example, FACS and flow cytometry are very much mainstream ways to profile cells. And these do give you single cell level information, but this is a serial measurement, right? You take a fluorescently labeled antibody, you put that into a collection of cells. The antibody labels the cells according to levels of the protein that antibody is specific for. And then you run the cells through a fax machine or a flow cytometer, and that instrument looks at one cell at a time. It squeezes the cells through an opening that's really just the size of a single cell, which also introduces a lot of stress. And it's pretty slow. I mean, 50,000 cells per minute may sound like a lot, but when you need to do a billion, that takes a while. Can take 2 weeks of sorting time. And the types of CRISPR screens that I'm going to tell you about, there are billions of cells. And so that's really 2 weeks of dedicated sorting time on one of these instruments, which is actually pretty hard to come by. So the approach that we've been working with, it's certainly modeled on flow cytometry. We refer to this as magnetic ranking cytometry. And instead of using antibodies that are labeled with a fluorophore, we use antibodies that are labeled with a very small magnetic particle. And so what we do in this type of cytometry is we use the magnetic particle to separate our rare cells from the cells that we don't really care about profiling. And then this approach also counts the magnetic particles on the surface of the cell in order to measure protein expression. And the way that we do that is using these microfluidic devices. So one of them is shown here, kind of in the middle of this slide. And if you look really closely at this fluidic device, so it's got an inlet, so the collection of cells are coming into the device and then flowing along the channel. And what you can see is that the channel is quite shallow at the beginning, and then it gets deeper and deeper. And so what that causes is a linear velocity gradient. And it turns out that the cells that have lots of nanoparticles attached to them, they will stop in the early zones of the device because there's a lot of magnetic force trapping those cells. There's magnets above and below the channel. So cells that have a high level of expression get caught in the early zones of the device. And then the ones that have lower levels of labeling keep going. And so the cells basically just array themselves along the channel according to numbers of magnetic nanoparticles and according to protein expression. So we can then come in with a collection or a cocktail of dyes or other fluorescent antibodies, stain the cells so that we can visualize them. And then we scan along the channel and find these cells, tabulate them, create the kind of histogram that's shown at the bottom of this slide. And again, this is single cell level information. And the distribution, it tells us a lot about the heterogeneity in terms of protein expression levels. It can tell us about multimodal distributions. It also gives us a count of very rare cells. So this is magnetic ranking cytometry, and what we think of as like its analytical mode, where we're just trying to measure protein expression for very rare cells. And this, just to show you some actual data, here we're looking at the protein EpCAM, surface protein that's found on epithelial cells. We have 4 different cell lines here. This inset is profiling of, I think it's 10,000 cells using flow cytometry. And the 4 different cells, you can see there's 4 different levels of EpCAM. Now, if we take 100 cells and we profile them using magnetic ranking cytometry, you can see that the data looks quite similar. But again, we're able to use a much lower number of cells to get this information. We can also look at multiple markers. So this technology is not that great for multiplexing because magnetism really only comes in one color. But we can split cells, run aliquots, and then be able to look at different protein targets. And so that's maybe a weakness, that we can't look at as many proteins as let's say a flow cytometer could. You can do kind of fast serial sorts, so that helps. But where this really provides a capability that you don't get from existing flow-based methods is the ability to go down to such a low cell count. So this is 10, 20, 40 cancer cells spiked into a milliliter of blood, where you have, again, billions of normal blood cells. And you can see that protein expression measurement stays very constant. And so we've shown that we can use this type of approach when we have a protein on the surface of the cell. That's pretty straightforward. But we can also fix and permeabilize cells. And then instead of having antibodies on the particles, we can use nucleic acid probes. We can target messenger RNAs and do RNA expression profiling at the single cell level. And then we can also do intracellular proteins as well. So that's the analytical version of the technology. What's also turned out, and this was a bit of a surprise, to be quite powerful is a preparative form of the devices that I showed you. And so here, instead of having that continuous channel, we have discrete compartments. And the compartments, we do our isolation of the cells, and then we can grab the compartment that contains the cells of interest and walk away with those cells and sequence them or characterize them or expand them. And what's really differentiating about this versus other ways to separate cells is that you can process about 1 billion cells in an hour. So that is a very large increase in throughput versus things like FACS. The recovery levels are very high and the viability levels are very high. I'll also show you a different device that works in a slightly different way. And this one, instead of having these compartments, we actually create a device that has magnetic deflection guides. And so these guides are made out of a magnetic material. It's called met glass. And so we pattern these so that the solution is flowing through the device. The cells, if they have magnetic nanoparticles and they're magnetized, they'll follow the guides. But at some point, the drag force actually overwhelms the magnetic force, and the cells drop off of the guides, and then they drop into these channels, and then we can get our subpopulations that way. So whether you use the device I just showed you or this device, I find people in my group kind of like one technology or another. One, I think, is a little bit better on throughput. The other one's a little bit better on the specificity and purity. So it depends a little bit on the application as to which configuration you use. But these videos just show you that, the cells are, again, they're clinging to these guides on the left as they flow through the device. And then on the right, you can see that at some point, the guides are turning, the drag force is going in the other direction. And so the cells eventually, they just drop off the guides into these different channels. And so the different channels then will contain cells with differing levels of protein expression. And you can do 10 channels, you can do more. It really just depends on the resolution of the sort that you're looking for. So those are the technologies. I'll just talk briefly about some applications, and I'll spend the rest of the time on one application in particular. We've done a lot of work using this approach over the years, looking at new ways to do liquid biopsy. Obviously, right now, looking at cell-free DNA is becoming more and more mainstream. We've worked instead on the side of things where we're actually collecting intact cells to gain information about the molecular level properties of a tumor. And so we can isolate tumor cells out of the blood for certain types of cancers. We can also isolate rare immune cells out of the blood. And we showed in a study of mesothelioma patients that that's actually a really interesting way to differentiate people that are asbestos exposed versus those that have developed mesothelioma. So I think there's actually a lot to do in that area to really figure out what immune cells are telling us about the disease state of a patient. We've also used this approach to do things like antibody selection. So taking large libraries of antibodies, incubating them with cells and a heterogeneous collection of cells, and then being able to pick out the cells that are of interest that have the antigen that we're selecting the antibody against. And you can then sort through a library of antibodies in basically one round and isolate very high specificity binders to a cell displayed target. We've also used this for cell therapy development. We actually use the ability of this approach to recover very rare cells, to go out and find tumor reactive cells in the blood. We know that there are tumor infiltrating lymphocytes that you can visualize within tumor tissue. We're able to use this platform and actually ask if those cells migrate out of the tumor into the blood. And we were able to find them and isolate them and show their anti-tumor activity. And so that's an area we actually just launched a new company that's going to be developing that as a therapeutic approach in terms of being able to harvest cells from a patient's blood, expand them, and then package those into a dose that could be delivered to a patient as a treatment for solid tumors. Also on the cell therapy side, we've used this as a tool to do quality control for cell therapies, because it's very important, especially for cell therapies that are derived from stem cells, that you don't have any leftover stem cells in a prep of cells that you're going to give to a patient just because of their tumorigenic properties. We've also used this approach to look at aptamers, kind of along the lines of what I described for antibodies. And then finally, we've turned this into an approach to do therapeutic target discovery. And that's what I'm going to spend the rest of my time on today, is talking you through how that works. And this area, so this is really an effort that marries the cell sorting approach that I just described with genome-wide CRISPR-Cas genomic screening, and really doing that on a phenotypic basis. So most CRISPR screens that are done today, and this has been the case historically, are based on viability. So you CRISPR edit all of your cells, you might stress them somehow, and then you're basically looking at the cells that survive the screen, sequencing those, and then looking at the ones that dropped out of the screen. And so your readout is really viability. It's kind of a yes, no type of readout. But what's even more powerful is selecting cells that have been CRISPR edited based on a phenotype, right? Did the levels of expression for a protein go up or down? Or did a cell maybe start secreting more cytokines or having lower levels of secretion? There's many other phenotypes that you could think about targeting. But again, this is pretty challenging because, a CRISPR edited library of cells to properly represent all of the guides and have enough redundancy in the library is about 1 billion cells. And so that's a lot of cells to handle to do a phenotypic assessment. So we put together a program, kind of a set of projects a few years ago now that we refer to as Leapfrog. And again, this is the marriage of our microfluidic platform with whole genome CRISPR-Cas screening. And so this program, Leapfrog, the acronym it doesn't catch all the letters, but it resembles large-scale phenotypic functional genomics at scale. And what's really powerful is that you can take an entire CRISPR screen, you can process all the cells in 1 hour. So you can take that screen through one of the devices, process it in an hour. And what this allows you to do is really scale up. So as I mentioned, if you want to do this with a fax machine, you're looking at about 2 weeks of sorting time. So that means you're typically going to do one cell line, one time point, one replicate. It's going to be a very focused screen. But with this technology, which is it's a very small cassette, we've been able to line up, many of these devices all on a bench. That's what's kind of shown on the right-hand side of this slide. It's just a bench that's loaded up with these devices. And so we can do panels of cell lines, panels of targets, multiple time points, lots of replicates. And what we found is that this allows us to produce really high quality data sets that validate very, very well and are able to provide us much more multidimensionality in the screen to make these types of things more informative. And I'll explain what I mean by that. You can also use fragile cell types, cells that won't really make it through a fax machine will come out the other end of these cell sorters with much higher levels of efficiency. So within Leapfrog, we have a few different efforts, and I'll touch on several of these today. We have a project that's focused on undruggable proteins and finding regulators of undruggable proteins. And there we're focusing on things like mutant KRAS and finding new ways to take that down in tumor cells. We have a series of screens that are focused on diseases that are either autoimmune or are really kind of based on a hyperactive inflammatory response. We've worked on immune checkpoints. We have a collaborative project with Amgen on antibody manufacturing. And then we've also taken some of this in vivo. So I'll walk you through several of these efforts. And again, mainly as food for thought about screens that maybe you have in mind, things that you'd like to be able to do, but you can't do using existing tools. And so I'm going to start with an initial study that we did just to benchmark this. And this was a paper that came out a few years ago. And again, basically the idea is that you take your guide library, you transfect that into cells, you make sure you have enough cells to represent all of the guides with lots of redundancy, and you let the guides then take effect to start maybe altering the phenotype or changing protein expression levels. We sort the cells and then we put them through a sequencing and validation pipeline. And the target that we decided to go after here is CD47. So CD47 is a protein that's attracted quite a bit of interest over the last few years. It's considered to be a don't eat me signal, right? You find it on normal blood cells, but tumor cells have found a way to hijack CD47 overexpress it on their cells. And what that does is it allows them to evade components of the immune system like the macrophages. And macrophages go around, as you may be aware, and they use SIRP-alpha to check the cell surface for CD47. If they find CD47, they leave the cell alone. If they don't see it, that's when their cytotoxic activities or the ability to do phagocytosis kicks in. So the thought has been for quite a while that maybe we could take CD47 off the surface of tumor cells or inhibit that interaction somehow and have a way to go after tumor cells. And this has been looked at by a variety of different biotech players, typically using antibodies to just block the interaction. And this has gotten a little bit of momentum in the clinic, but has not really been effective in solid tumors. And so we thought this was an interesting target where we could go in with a phenotypic screening approach, look for alterations in what was going on with CD47, and then maybe find regulators of CD47 expression. That was the idea. I'll tell you, as we did the screen, that we came out with a different set of conclusions. But let's find a way to disable CD47 through something other than just a direct blocking effect. And so we did this screen, and we did it because it was the first kind of trial of this platform. We did it head-to-head with FACS and looked at the hits that were common between the two cell sorting technologies and came up with the results shown on the right. So you can see that CD47, those guides showed up as the third hit in this screen, which you should always see guides for the thing you're trying to knock down come up in your screen. So we see that, which is great. And then the top two hits, even above CD47, one was a solute carrier, the other was a protein called QPCTL. We decided to go after QPCTL because it's an enzyme, and so this gives us something that is easier to go after as a drug. And I'm just trying to move my pointer here for a minute. Okay, so we chose QPCTL as the top hit here, and this protein, its function in cells is to cyclize glutamine to form pyroglutamate. So it's a cyclization reaction. At the time, there was no known association between CD47 and QPCTL. So if you put those two things into a search engine, you just got a blank page, which is, you're all aware, is quite unusual. And so we really had to kind of start solving this puzzle as to what was the connection between these two factors that had this enzyme come up as a top hit in our screen. And it pretty quickly emerged that, interestingly enough, CD47 has a pyroglutamate at its N-terminus. So it has this modification at a very important interface on CD47 because this is where the protein contacts SIRP-alpha. And so the presence of the modification made us think, well, maybe that's the link with QPCTL. Perhaps this enzyme is cyclizing the N-terminus. And we realized then that maybe our screen was actually not reading out expression levels for CD47, but it was actually reading out modification levels because of the antibody that we chose. It just turned out the antibody was specific for the modification. So this ended up being a modification screen rather than an expression screen. But the moral of the story is you have to be careful about what antibodies you use. But anyway, we went on to validate QPCTL as something that did modulate this modification in CD47. And so we were able to show that. And again, this was a really striking finding because this N-terminal pyroglutamate is right in the binding pocket that SIRP-alpha is using to look for CD47. So it's a really critical interaction. And so we were able to, fortunately, find an inhibitor for QPCTL. This is an enzyme that's actually been looked at in the context of kind of what happens in plaques in the brain. And so there were some compounds that were around that had been looked at in the context of Alzheimer's. And so we were able to grab this compound, use it to inhibit QPCTL. And then you can see that, so this compound is SEN177. And so when we dose cells with SEN177, the antibody that binds the pyroglutamate modification, it no longer binds. And so we can see that we're able to drug this modification using something, completely orthogonal to CD47. And so again, this is the idea that we find regulators or modulators of things that could be therapeutic targets. And so we're now expanding on this. We're looking at new checkpoints, and also we have teamed up with Evotec on finding new compounds that will allow us to go after QPCTL as the beginning of a drug development effort. So that's kind of how we got started on this effort. And so now I'll tell you about some newer projects that I think most of the ones that I'm going to tell you about are actually unpublished. Some of them are not totally finished, but I think the data is at a point where it's interesting to talk about. And so one thing that we decided to go after was looking at mutant KRAS, right? This is a type of protein or a modified protein that is found in many, many different tumors. There is a spectrum of different mutants that appear in different types of tumors. This has also been an active area recently with companies like Amgen going after the G12C mutant. And the way that they did that is by developing chemistry that is specific for that cysteine residue that's only in the mutant protein. Cysteine has a reactive side chain, which is a thiol moiety. And so a little bit easier to develop specific chemistry than, for example, if you were going to look at the G12V mutant that basically just has methyl groups in the place of other methyl groups. So not as amenable to that same type of strategy. And so we decided to kind of dig in and with this screening strategy, go after specific modulators of the G12V mutant with an eye towards the fact that in pancreatic cancer, colon cancer, lung cancer, that there are significant levels of the G12V mutation found in patients with these solid tumors. And the trick here is to really affect the mutant form of the protein specifically, right? Wild type KRAS is really important for the function of the cell. And so we need to be able to find a way to modulate the mutant very specifically. And so we performed a screen here, and this screen was done so that we were looking at cell lines with different genetic backgrounds. And so, for example, we looked at a cell line that had 2 wild type copies of the KRAS gene, and then compared with a cell line that had 2 copies of the G12V mutant. And then compare the data, and you can see that there's some hits that kind of pop out on the wild type KRAS side of things, but then a completely different set of hits that pop out on the mutant side of the screen. And so this is the type of analysis that really allows us to look specifically for modulators of the mutant. And I'm not going to talk too much about the specific biology. We're still dealing with IP and that kind of stuff. So I'm just referring to this as SMKM. This is an enzyme that's involved in lipid metabolism, kind of an unexpected link, something that the biology we're still kind of characterizing and understanding. But what we were able to see is that we could validate this quite straightforwardly, so that if we knock SMKM out of a cell line, we see about 90% reduction in mutant KRAS levels. And so a very significant lowering of this mutated protein. And we were also able to kind of follow that up with showing a proliferation defect. So again, if you go into a wild type cell line and if you knock out SMKM, you don't really see any change in proliferation. But if you go over into a cell line that has the G12V mutant, and these cell lines are typically addicted to mutant KRAS, right? And so there, if we knock out SMKM, this proposed modulator of mutant KRAS, you can see that there is a very significant proliferation defect. And so this is showing us that we were able to achieve this type of specificity that we were looking for. We've been looking at this in vitro, also in cells, also in mouse models. We were fortunate to find a probe compound for this particular enzyme, and so are able to kind of follow the druggability in different model systems, and this is something that we're going to take forward once we really have to nail the mechanism. I think that's the most important thing in this project. So then I'll move on to another type of screen that we've been working on that relates to the function of T cells. And I think for this audience, I don't really have to tell you about how T cells work, but basically very important in the tumor killing context. The T cells recognize other cells, tumor cells, and then are able to release cytokines and other toxic agents in order to eradicate tumor cells when things are working well. But this obviously can kind of go out of control, right? And we do have many diseases that seem to be driven by T cells recognizing cells that are actually healthy, and then really causing a reaction stemming from this immune reaction within one's own tissues. And so we became interested in trying to adapt this screening capability to look for ways to tune down cytokine secretion. And so to maybe provide a way to look at modulating autoimmune diseases or inflammatory diseases. And in particular, we got interested in thinking about this within the context of things like IBD, and focusing on the cytokine interferon gamma, which in inflammatory bowel disease seems to be kind of over-secreted, and that over-secretion ends up destroying the intestinal stem cells. And then that is then the basis for the physiological effects of IBD. So this screen, it required that we develop a new capability, because I talked to you before about screening based on expression or modification. Here we want to screen based on secretion. We want to take a library of edited cells, we want to allow them to secrete, and then we want to separate the cells according to secretion efficiency. And so what was done, and this was the work of Mahmoud Labib, who's a research professor in the group, who put this all together. And Mahmoud came up with a way where he actually parked antibodies on the surface of cells, of T cells, and then the secretion would take place, and then the antibodies would catch the cytokines on the surface of the cell, where he could then label them with magnetic particles and then use our cell sorters to do the same kind of thing that I showed you previously. Here we are working with primary T cells, and so it's a little bit harder to go with a full genome-wide screen here. So this first screen that we've done on a secretion basis was just done with kinases. And so when we did this screen, we ranked the kinases that validated well. We then went into T cells and knocked these genes out or down, and then measured interferon gamma secretion. You can see that the results validated very well with really low levels of interferon gamma secretion, especially for the targets kind of shown on the right hand of the plot. And then we started pulling small molecules that were known inhibitors of these kinases, and started dosing the cells to see if we could lower interferon secretion through the action of a small molecule, and that worked quite well. And so we focused in on this compound, XMU-MP1, as one to take forward into an animal model. And we're able to, and this was in collaboration with the group of Stéphane Auger at the University of Toronto, we were able to test this within a rat model of IBD. And basically the way that this model works is that you trap treat the rats with a chemical that induces the symptoms of IBD. And so we have a control group that we just treated with tap water. We have the group that we treated with DSS. And then we have another group that we treated with DSS, and then we treated with our kinase inhibitor. And the cleanest way to then really look at this is to by visualizing these intestinal stem cells that I mentioned end up being damaged by too much interferon gamma secretion. And so you can see these intestinal stem cells quite well in the tap water cohort where we're not inducing IBD. So they're these little brown structures here. Then if we have the cohort of rats that were treated with DSS, you can see that those stem cells are gone. And now if we treat with our compound, you can see that we're able to rescue the intestinal stem cells. And so this, provides a new way to go after these types of inflammatory phenotypes. And this is something that we're excited about really extrapolating to different cytokines, different models. And so another area that we're very active in kind of going forward. And then I'll just mention briefly that we've also worked with Amgen on a similar type of secretion screening, where here we're looking at the secretion of therapeutic antibodies and those proteins coming out of cells. Same approach, we catch them on the surface of the cell and label them. And we have a really nice screen where some interesting hits have emerged that allow us to hopefully in the future develop cell lines that are really super secretors for this type of therapeutic application. And so finally, in the last few minutes here, I'm just going to tell you about the application of this type of approach to an in vivo screen. And this screen is one we've had a long-standing interest, as I mentioned, in liquid biopsy and looking at tumor cells in the blood. And so this screen focuses in on those cells. So these are cells that break off from a tumor. They enter the bloodstream, and then eventually they exit the bloodstream at a site where they will then seed metastatic tumors. And this is really the reason that cancer is so problematic, right? Because of the formation of metastatic lesions and their effect on different organs within the body. But this process is really a bit of a black box. We don't know a lot about why the cells leave. We don't know what really promotes that. We don't really know what promotes them to come back out of the bloodstream. So a lot of things to learn here. And so this screen was actually the idea of postdoc in the lab, Yuan Ma, and a graduate student, Fan. And Yuan and Fan came together and decided to give this a try. It was kind of a pie in the sky idea where they thought what they would do is to CRISPR edit cancer cells that they would then put into an animal, allow tumors to grow out of these edited cells, and then they would wait for those tumors to start shedding cells into the bloodstream, and then do the sequencing, look at what knockouts were really promoting the levels of these circulating tumor cells in the blood. And this was a very challenging screen because they were just getting maybe hundreds of cells from their mice in order to do the sequencing analysis. So they had to do an initial screen, they had to come in and then do a sub-library just to really get the statistics and the bioinformatics to work well. And this was just published a couple months ago in Science Advances. They found this really interesting factor SLT2 as really the top hit in the screen. And then they were able to show, I mean, when you knock out SLT2, that does cause a tumor to grow faster, but it also increases the levels of circulating tumor cells. Even when you have tumors that are the same volume, you'll see the tumors with the SLT2 knockout releasing many more cells. And then they were able to show that knocking out SLT2 really did promote metastasis. They could see infiltration into the lymph nodes and then levels of metastasis that were much, much higher in the knockout mice versus the wild type mice. So this is, again, an area that we have many more projects that are going forward in. And the way we think about this is that as we've tried to discover and develop new therapeutic modalities in different diseases, we typically go after, right, the enzymes, the active sites where we can go in with a small molecule, or we go in with an antibody that can be a binder or a blocker. But there's all this stuff under the surface, right? There's all of these regulators that we could go after to provide new opportunities for therapeutic development. And I'm going to end just with something that's maybe a little bit more on topic, just in terms of something that we've been thinking about closer to the interests of this group, in terms of applying this type of screen potentially to something like IgA nephropathy. And could we apply this to a phenotype that's almost more of an adhesion phenotype or an aggregation phenotype? And we think that that's something that's very doable. So we're still kind of brainstorming about this, and talking to people like Sue about how we could potentially get this off the ground. Again, we focused mainly on the cancer side of things, just maybe that's where we have more expertise than in other disease areas. But we're very excited to collaborate with people that have their own, favorite disease phenotype, that this type of approach could maybe help them uncover some new biology. So with that, I'll sum up here, and I really want to thank my group. Everything that I told you about today is the work done by a really brilliant collection of graduate students and postdoctoral fellows and research professors. And in particular, I want to call out Randy Atwall, who was in my group at the University of Toronto and moved with us to Northwestern, and really runs that leapfrog effort that I told you about kind of as a whole. And then Mahmoud, who I mentioned, that worked out all of the cytokine secretion screening. And then Gloria and Abdallah are two graduate students also working in this area. So just wanted to specifically thank them, and thank you again for having me and listening, and I'd be very happy to answer questions.
Thank you so much, Dr. Kelley. That was beyond amazing. And I am sure that, everyone in the audience is just like thrilled and excited with the potential of your work in our field. So we will have some time for questions. Would your schedule allow any extension in case that it's needed?
I have 10 minutes till my next meeting. I'm kind of in back-to-back meetings all day today, but I'm always happy to be contacted, by email and chat offline.
Excellent. And while we are waiting, for questions from our audience, I will make, the start. And I was wondering, with your magnetic cytometric protocol, if there are any dissociation bias in terms of the cells that you can capture. And the other question is, do you need to apply this platform on fresh or frozen tissue? I'm just basically thinking ahead of the time for human biopsies, etc.
Yeah, so it works with either frozen or fresh tissues. They just need to be dissociated. We have also worked with fixed cells, so that is a possibility as well. Cells that are hard to dissociate can be a challenge, so you do need to push the dissociation side of things. But we've worked with a variety of different primary cells. We're not very sensitive to cell size. So cells that would be problematic to put through a fax machine because of their size, or I think I mentioned this because they're kind of fragile and seem to have viability issues in a fax sorter, those types of cells work pretty well with this platform.
Excellent. And I think that this answers possible the question of Dr. Quaggen about many cells in the kidney being difficult to work with and requiring gentle sorting. So probably, and you can comment, your platform seems to offer an advantage.
Yeah, it is very gentle. So again, like a fax machine, you squeeze a cell through an opening that is only the size of a single cell. And that creates a lot of shear force and just it really beats the cells up. in our devices, we're just flowing through a channel and we're just kind of letting the cells like, self-organize based on the particles that they're carrying. So we've looked at levels of things like oxidative stress, obviously viability, and that is much, much higher in our cells. Even more so for our approach, there's another magnetic separating technology that's commercial, it's used very widely, it's called MAX. That also seems to stress the cells because you kind of put them through a column and there's just more force on the cells, whereas ours, they're just kind of, flowing through pretty gently. So I do think it's quite a good fit with fragile cell types.
That's amazing. And the cells that you have basically selected, can you actually subject them to other platforms following, for example, single cell? Let's say that, beyond imagination.
Yeah.
Any questions from our audience? We have like few more minutes. I don't want to monopolize the discussion here. Well, I think that probably it's just too many questions to ask. We have, in the setting of kidney transplantation, immune cells activated that can be assessed what they secrete or targeted therapeutically, genetic diseases. We can assess regulators as we propose. It's just amazing. I cannot even comment more on that. So if we don't have any more questions, I think that I will close the sessions and with some
Penelope?
Yes.
Penelope, can I ask a quick question to Shana? It's Sue.
Go ahead.
Yes.
Okay, sorry. Yeah, Shana, that was fantastic. And I know just too many ideas trying to all the potential applications in the kidney space. The cytokine secretion is fantastic. And so one question, that I see a big issue in the kidney space is secretion of, it's not necessarily cytokines from kidney cells, but extracellular matrix molecules and various things. Is it feasible to adapt to other secreted molecules beyond, those that might cytokines?
Absolutely. Yeah. Anything, as long as there's a binder out there, right? Like some kind of an antibody, something that we can park on the cell so that as it's being secreted, we can catch it. Yeah. Anything can be used the same way.
And one thing I hadn't recognized is that I didn't realize you could actually take whole tissue, dissociate, and actually subject to this technology, which really opens up a huge, I think, opportunity. And so something that I'll need to follow up with you after the seminar. I think that's, opens up incredible opportunities for our field.
Yeah. Well, we'd love to talk more about it soon.
Well, that's really amazing, and we have many comments, in our chat box, echoing, the potential of your work in our kidney field. So thank you so much for joining us for today's NOCKS seminar. Please follow up NeverHub at Twitter, and keep in mind Dr. Shelley is available in Twitter at Shana Kelley, so you can follow up updates from her amazing work. And thank you, everyone, and you have a great day.
Thanks very much. Bye-bye.